Hello, everybody, and welcome back to BioSC 140 Human Physiology. This video is the discussion video for Lab 4 Thin Layer Chromatography. A little review. Proteins are polymers. Polymers are molecules that are made up of many small building blocks. Each of one of those individual building blocks is called a monomer. The monomer for the monomer for proteins are amino acids. Amino acids are the building blocks of proteins. You connect a bunch of amino acids together and you produce a protein. All amino acids have a similar structure. All amino acids are going to have an alpha carbon. Off of that alpha carbon, all amino acids are going to have an amino group. The amino group is where amino in the word amino acid comes from. They're all going to have a carboxyl group. The carboxyl groups are acidic. That acidic carboxyl group is where the term or the acid comes from, so amino acid. Off of that alpha carbon, you're also going to have a hydrogen, and you're going to have something called an R group or a side chain. That R group or side chain is what varies in between different amino acids. So different amino acids will have different side chains. You can see there's a whole bunch of non-polar side chains. There's a whole bunch of polar side chains, and there's acidic side chains and basic side chains. So the chemistry, the chemical properties, the charge, the polarity can vary amongst side chains or R groups. Also, what else do you notice is different? The shapes. Form and function matter. The shape of the amino acid will impart different characteristics. The shape of the R chain, of the side chain, will impart different characteristics upon that amino acid. So some side chains are small, some are large, some have rings in them, some are branched. There's a lot of different structural shape characteristics to the amino acids also. Some are larger and heavier, some are smaller and lighter. Now, let's review what we did for this lab. Now, you should have watched the previous video where we really go into exactly what we do. I'm going to give a quick overview of what we do in this lab. We have a plate called a TLC or a thin layer chromatography plate. One side is glass-like, one side is a little rough, kind of matted, like sandpaper. On that plate, about an inch above the bottom, we're going to draw a light pencil line. We're then going to put 2 little drops of alanine on that line, 2 little drops of aspartic acid right next to it, 2 little drops of histidine next to it, and so on, all the way down to our 6th spot where we're going to put a few drops of an unknown amino acid. We're then going to take that plate and put it inside of a chamber that looks kind of like a fish tank. And at the bottom of that fish tank, we're going to have about a 1/4 inch or 1/2 inch of the solvent. The solvent is 50% water, 50% ethanol. And that solvent is going to move up the plate. This is similar to if you take a paper towel and you dip that paper towel in water. That water is going to latch onto the paper towel, and you'll see it kind of creep up the paper towel. Same thing is going to happen here. That solvent's going to move up the plate. And as that solvent moves up the plate, it's going to encounter the samples that we put on the plate. And those samples are going to get pushed and pulled by the solvent up along the plate. After we've allowed for enough time for the solvent to move a majority of the way across the plate, we're going to remove the plate from the chamber. And we're going to draw a light pencil line across where the end of the solvent is, however far the solvent moved. A light pencil line. At this point, our amino acid samples will have been pulled varying distances along the plate. I'm going to give you that little heads up. The different amino acids will be pulled different lengths, different distances along the plate by the solvent. Once we have that line drawn, we're going to take this plate to the back of the lab room and we're going to spray it with something called ninhydrin. Ninhydrin is carcinogenic, so it's actually me who puts on the safety gear and sprays the plate for processing. We're then going to move the plate and put it into an incubator or a warm oven-like chamber to help it dry quickly. Once the plate's dry and sprayed with ninhydrin, we're actually going to have these amino acid samples are actually going to, we're going to be able to see them. They're going to have a color. We're going to be able to visualize those amino acids. We're going to have a dot here. We'll have dots all over the place, actually. What I want you, so after we see our dots, what we're going to do is we're going to look for something called the relative front. So the RF ratio for each one of these amino acids. So to find it, the RF is equal to D for the S, so distance for the spot. So we're going to measure the distance from the starting line to however far the dot went for each one of these amino acids. And we're going to divide that by the distance of the front, or the total distance that the solvent traveled. And what we're going to do then is we're going to compare the RF value for each one of these amino acids with our unknown. And we're going to try to figure out which amino acid is our unknown amino acid. If the RF values are equal, if the RF value for alanine is equal to the unknown, there's a good chance that alanine is our unknown amino acid. Now, I want you to do some thinking before we move on to the next slide. What might affect how far the spot travels on the plate? What might affect how far the amino acid does move or doesn't move on the plate? What might affect how far the amino acid moves on the plate? Think about amino acids. Think about what we talked about at the intro to this video. Think about what you know about amino acids. What might affect how far the amino acid moves on the plate? Well, thin layer chromatography separates amino acids, and it separates amino acids based on the R group size and chemistry. Size and chemistry. Migration, how far the amino acid travels, depends on the solubility in the solvent. So as the solvent passes over the sample, the amino acid sample, there's some amino acids will be attracted to the solvent, and the solvent is actually going to pull that amino acid along with it. The solvent that we used was 50% water, 50% ethanol. Water is a polar molecule. Ethanol is a polar molecule. Polar molecules are attracted to other polar molecules. Polar molecules actually repel nonpolar molecules. They're not attracted to each other. Nonpolar molecules are attracted to nonpolar molecules. So like dissolves like. So the solvent pulls amino acids up the plate. Polar amino acids move further in polar solvents because they are more soluble. The more soluble the amino acid is, the stronger the attraction between the amino acid and the solvent, the further it's going to get pulled along the plate. Another factor is the attraction to the plate. The plate actually has a little bit of polarity to itself also. And the plate is going to grab onto those amino acids and actually hold them back. So the stronger the interaction between the plate and the sample, the slower the sample will move. So those are kind of contradictory forces. The solvent's going to promote migration, promote movement, and the plate will actually slow down motion for polar molecules. The other factor is molecular weight and size. So larger molecules migrate more slowly. Larger molecules migrate more slowly. The way I rationalize this, I understand this, is it easier to pull something large or is it easier to pull something small? Molecular size, large molecule matters. So, will two amino acids have different RF values? The two amino acids have different RF values. Theoretically, yes. Practically, not always. So every amino acid has a little bit of different chemistry, size, and shape. And theoretically, if you let the process run long enough, if your measurements are accurate enough, every amino acid will have a different RF value. What happens in lab, though, is sometimes amino acids are really similar. They're only different they're one carbon different. They're very similar. And what can happen is we don't let the process go on for long enough. And so it appears that two amino acids have the same RF value, when in reality, their RF values might be 0.001 different. It's just using our rulers, our tools, we don't have enough accuracy for it. So, let's say amino acid 1 moves 2 inches and amino acid 2 moves 2.0001 inches. Practically we might not see that difference in lab. What influences our resolution? What could we do that situation I just described where they moved almost the exact same distance? What could we do to make that difference more clear? Well, we could allow the process to go on for longer. We get a bigger plate. If we allow the plate to run twice as long, that difference is going to be twice as noticeable. So the further a solvent runs, the better the resolution, help distinguish between different amino acids. Can we compare RF values between different TLC experiments? Well, theoretically, as long as the solvent and plate are the exact same, the RF value should not change. The migration distance will be different, but it should still be the same relative distance. So provided you use the exact same samples, exact same amino acid, exact same solvent, the RF values should stay constant, even if in different runs of the experiment, you let the RF travel the same, different distances. So, if your DF is right here in time 2, and right here it's time 3, the RF value should still be the same. So right down here, we actually have one of these plates. We actually have one of these plates down here. This is what it looks like when we finish in class. So what other characteristics do you notice? What else, what varies, what is different between each one of these samples? Shape and color is actually different, and you can actually use that to help you identify what the unknown sample is. All right, so I want you to know the background information about the amino acids. I want you to be able to tell the general story of what we did for the TLC experiment. I want you to know what affects how far an amino acid travels on the TLC plate, and I want you to be able to calculate this RF value down here. This RF value down here. With regards to the RF value, practicing makes perfect. There's sample data in your lab book in the supplement, and there's also a really good worksheet that I posted on Canvas. The worksheet that I posted on Canvas will walk you through a few of these RF problems and will get you prepared for the checkups and get you prepared for the quiz. If you have any questions on it, please let me know. And until the next video, have a great day.